that amazing introduction. I'm super happy to be here with you all virtually this evening. I hope to be with you all in the forest at some point in this life. I'm sure I've been in the forest with some of you all jumping around these areas. And I do implore you all to join us at MycoFest this year. Not too far off from you guys. We're going to be doing it in Tamaqua, Pennsylvania this year. The land of the boletes. It's incredible over there, and I hope to experience it with some of you all. So yeah, I'm here to talk to you guys today about Cordyceps cultivation, Cordyceps culture in the United States. And when I say Cordyceps culture, I'm not talking about in a petri dish per se, but the social culture growing around Cordyceps mushrooms in the United States has been very interesting to be a part of, to witness, and to be living in. So we'll hit a little bit on the culture of Cordyceps in the United States, and we'll cruise through cultivation. Cultivation of Cordyceps. I'm about to teach a 2-day course on it this weekend. I could talk for 2 whole days about cultivating Cordyceps and all of the things associated with. But we'll go through a cool little overview. So welcome to the CordyQuest. I always introduce everybody to Cordyceps with the CordyQuest. It's a journey I've been on for 7 years now. I was introduced to Cordyceps mushrooms through public broadcasting or public access television in my late teen years. There was an enthusiastic gentleman selling magic bullet blenders on a infomercial. Late at night, I had the TV on, and he was talking about throwing superfoods in the blender and this medicinal mushroom and medicinal mushroom tea. And I'm like, what is this? This sounds amazing. And it was like a point in my life where I was focusing on nutrition. So I was introduced. I think that was how I was introduced. And then I came across the mushrooms in a bag of Chinese Cordyceps mushrooms back in like 2015. And then at our first MycoFest, we found wild specimens. And here in Pennsylvania and even in New Jersey, we have native Cordyceps militaris. It's the most common Cordyceps that we'll find in the wild here. And it is prolific in the Appalachian Mountain Range. So super lucky to be in an area where I can access the wild Cordyceps. In the picture on the right there, the Cordyceps with the little brown on the bottom are wild specimens pulled off of an insect, and the ones on the right are cultivated indoors. You can see the ones on the left are a little bit roughed up from weather and all that kind of stuff in comparison to the soft and undisturbed ones on the right. So as we know, Cordyceps of the kingdom fungi, phylum Ascomycota. So it's an ascomycete like morels. But unlike morels, the Cordyceps have perithecium on the stroma where the asci are located, not in the I don't even know what's the technical term of the little holes on the morel where the asci are. So they have Sordaromycetes in the order Hypocreales, which contains a lot of really interesting parasites. The more as I did study and research for my second book is when I really started to realize how many interesting mushrooms are in the order Hypocreales or Hypocreales, or however you would say it. But it's interesting how that group of fungi evolved to parasitize not only insects, but other fungi and plants. So ergot is of the same order, and they're producing all sorts of interesting alkaloids, like LSA, and even the corn smut, same order, and all of the truffle parasites. And what I find the most interesting is that the compounds that these fungi produce in order to parasitize their hosts may be detrimental or deadly even to the hosts, but to us, it's very stimulating or interesting or has different effects on us in comparison to like an insect or a plant or something. In the family Cordycepitaceae, there are a lot of variations. Ophiocordyceps is where we have the classical Ophiocordyceps, like the Ophiocordyceps sinensis, that is told stories about in Tibetan culture, and a lot of people, if they have any relation to cordyceps in any mycological communities, it's usually through stories of Tibetan cordyceps until the cultivation of militaris in more recent years. I mostly work with Cordyceps militaris. As we start to expand our lab out and build more teams, we will be working with some of these other cordyceps, which I'll talk to you about. But I always love to emphasize that Cordyceps militaris and the cordyceps I'll be talking about are actually cordyceps and not Ophiocordyceps. Everybody loves to say cordyceps from Tibet, but it's Ophiocordyceps from Tibet. Why don't we call it that? So Cordyceps militaris is of the Cordyceps militaris complex, 4 mushrooms that look very similar to Cordyceps militaris. You know, I speak a lot for these scientific communities, and we have to get all technical with the different species and things like that, which is very beneficial for research and things like that, for curiosity. But for a general understanding, it's better to look at these as families and not get so technical with all of it. All of these are going to have similar medicinal benefits and pretty much be cordyceps. But in this world that we're living in now, it is beneficial sometimes to highlight the differences in these and the nuances in these things. So we have Cordyceps militaris, Cordyceps pseudomilitaris, Cordyceps roseostromata, and Cordyceps cardinalis. They all look macroscopically identical. They do have variations in hosts, but still will mostly grow on coleopteran and lepidopteran hosts, coleopteran being beetles and lepidopteran being moths and butterflies. And they can grow on the adult forms, on the pupal forms, the pupiform, and on the larval forms. We rarely see them on the larval forms and rarely see them on the adult forms, mostly see them on the pupa. And there's like, Cordyceps militaris is the most successful cordyceps in the world because of how many different species it can grow on, over 50 different species that we know of. So whenever I was doing my research in my first book, I put like 30 or 40 different species because I cited a research paper from Korea. When I wrote my second book, I went back to cite that same research paper, and then I started looking up all the insects that were listed, and only one of them was insect that is native in North America. All the other insects are from Asia. So I was like, wow, this research was done in Asia with Asian insects. So we haven't even dug or scratched the surface on the insect host species in the United States. So I sent a bunch of pictures to entomologists of different hosts that we were finding them on with not much success. We speculated and discovered that they were growing on Anisota senatoria, mostly in Pennsylvania. It's like an orange winged oak moth or orange tip oak moth. But still in my area, I see them on like 5 different host species. And I haven't ever confirmed having pseudomilitaris, roseostromata, or cardinalis. I very well may have, because we'll harvest like 100 cordyceps sometimes in a day or more. And I don't know. I'm not stopping and looking with every single one of them so closely to be able to tell that. But we could do the DNA now, which I'll talk about that more a little bit later. But another reason why this will be beneficial into the future, and it's something that we're starting to do now at Mycosymbiotics, is we're marketing reishi species. Like, kind of funny, it sounds like reishi species, but we're marketing reishi species because I noticed over time of consuming reishi or however you want to say it, that different species made me feel different. I don't particularly like Ganoderma tsuge. I like Ganoderma curtisii and I like Ganoderma sessile, but I don't like Ganoderma curtisii for drinking teas of. It doesn't feel the best for me, and my body makes me feel a little aggro. But Ganoderma sessile, Ganoderma curtisii makes me feel a little bit more calm. And so I noticed that in myself, but if whenever I didn't know any mushrooms at all, everything just tasted like mushrooms, and it was hard to like it was hard to understand how the experiences I was having. But it took 10 plus years of consuming mushrooms, and okay, like, oh, when I put reishi in my body, that's what's different, or that's how I feel. So it took a little bit of discernment, experiential discernment, to um, know that I liked these different reishi. And where I was taking that, or where I wanted to go with that before I went to the next one, is that as we can do this R&D with ourselves and in the team, as we get our hands on different types of cordyceps, as we get our hands on different strains, we can all sit with each other and consume tea. And how are you feeling? I'm feeling like this, or this is how I feel, or da 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 da, and like do that over time, because it's like you can't do that one time and say that this is what it is. You can't do that by yourself and write a description, olfactory, sensory description of it. You need more people because everybody's tastes are different, all this kind of stuff. So as we right now, if you give anybody cordyceps and they haven't had it, they're gonna probably not know, or be like, it all tastes like the same, or it all the experience is all the same. But as we start to be able to discern it, I think that these things will be highly valued as their own product. Like, okay, like we have our cordyceps militaris product, we have our cordyceps cardinalis product, and this one makes you feel like this one makes you feel like that. And as we've seen in the Hericium genus, Hericium erinaceus has those species-specific unique compounds, the erinacines in the Hericinones, but Hericium coralloides has 3 novel compounds in it that they're just calling corallosins 1, 2, 3, or corallosins A, B, and C. So we know that just in the Hericium genus itself, the different species in that genus are producing different medicinal compounds. Hericium coralloides is also producing neuroprotective, neuroregenerative compounds that act differently than the erinacines in the Hericinones. So I'm sure we'll discover this in Cordyceps militaris, just in the complex, but it hasn't been discovered. It hasn't been researched yet. So yeah, life cycle of Cordyceps militaris. It's really cool, these old pictures from the 1800s. The people accurately, with the cross-section microscopic depictions of the cordyceps, you can only imagine the wealthy, educated individuals with their microscopes just having a field day all day. Like literally, like one of the old 1800s, 1900s. I think it's the 1800s book that I referenced for illustrations. It's a Danish book, and it translates to something like "Fun Day in the Hills Looking for Mushrooms" or something. Like that's the title of the book, and I'm just like, oh, that's great. You know what I mean? Like just having a luxurious life out there picking mushrooms and looking at them under the microscope. So a little bit about the sex life of Cordyceps militaris. They are bipolar heterothallic. This means they need 2 mating compatible spores in order to produce fertile offspring. So when I'm doing this in the lab, when I'm breeding in the lab, I have to isolate spores, and then I have to figure out whether or not they're mating compatible. So because if I have 2 of the same mating type spore, mating type, you could think of it like a gender. So each spore has its own gender, and there has to be a boy spore and a girl spore to connect to make a baby spore that can produce offspring. 2 boy spores can come together and make mycelium, but they cannot make fertile mushrooms. 2 girl spores can come together, but they can't make fertile mushrooms. So and I can't tell if they're a boy or girl. They don't have any like genitals or anything like that I can look at to see. So I have to do DNA analysis. So what I'll do is I'll extract DNA from the spore after it germinates, and from the mycelium, and we will amplify it as the mating type genes, and then I can run a gel to visualize the results of my PCR or of my amplification of the mating type gene. And I should only get one or the other. Like if I have to test for both of them every time because there is a possibility. That's the other interesting thing about mushrooms. So like once the boy meets the girl, then it's both. It's not like a boy or a girl anymore. So like once the two mating types combine, then that mycelium DNA has both of the mating types. So if I'm testing the spore for mating types and it shows me both, that means I messed up and I didn't, and I already got two spores together on accident. So that's also really good to know when you're breeding, because then that can change up everything that you're doing. So you want to make sure that you only have one mating type showing, and you have to test for both of them to see. So after all of that science jargon, I figure out if they're boy or girl. I put them together, make more cordyceps that are really beautiful for commercial production. Cordyceps can also anamorph. This isn't something I've witnessed in the lab, but they can turn into a mold. They can turn into two different molds, Simplicillum and Lecanicillum. So this was like really mysterious to me for a while, until I met Daniel Winkler up at the Puget Sound Mycological Society. Yeah, that's the one I met him at. Yeah. And he does these mushrooming tours where he goes to different countries and takes people out to go look for mushrooms. And he's been going to the Himalayan mountains for years. And he told me that the Ophiocordyceps sinensis that they find on the caterpillars in the mountains there, it anamorphs into a endophytic fungus. Endo means inside, phytic, phyte means plant. So that Ophiocordyceps morphs into an endophytic fungus that lives in the leaves of the plant that the caterpillar eats, that it's hosted on. So it's so crazy when you think like this little mushroom smaller than my pinky is supposed to have two mating compatible spores that lands on one bug to produce fertile offspring, because like you have to have the mating compatible spores. So I'm just like, it has to have some other mechanism to be able to exist in the environment if it can't do that. By morphing into a whole different species. So Cordyceps militaris will morph into Simplicillum and Lecanicillum and literally change its whole sexual function from producing spores out of a ascus to producing conidia spores off of its mycelium. Super crazy. And then it just exists in the soil. So I'm like, I finally put it together. I'm like, wow, Cordyceps militaris morphs into a soil mold, probably because the insects bury themselves in their pupation phase, the insects that they grow on. So it puts themselves in direct connection with those insects. There's also a couple different things that we've noticed over the years about how they move through the environment. So if we ever find a Cordyceps militaris in the wild and I pull it and the insect is cracked, I won't take that insect into the lab because it generally will have little mites inside of it. And the mites will crawl in and out of your petri dishes and eat mycelium. And they're eating the mycelium inside of the bug, but then they're crawling around through the soil looking for more food, carrying mycelium or carrying the cells of the Cordyceps mushroom to different places. So that can be one means of transport. But also, I learned that, what is it called? Oh, I can never remember. When fruit ripens on a tree, and you knock against the tree, the ripe fruit will fall off, but the unripe fruit will stay on the tree. If you really, really shake the tree, then the unripe fruit will fall off. But there's like a adhesion zone or abscission zone. That's what it's called, abscission zone on fruit that withers away whenever the fruit starts to ripen, and fruit will fall off. I think that Cordyceps militaris has a similar thing with the perithecium on the top. I have some pictures. I can show you the perithecium a little bit better. But the perithecium will grow on the top of the stroma on the Cordyceps. They'll produce these little bumps. But when they're mature, the bumps will fall off if you just bump them. Like if you touch them with your finger gently, the bumps will fall off. And if you look under a microscope, it's like a little sac that's still shooting out spores, even though it fell off. So I'm just like a beetle walking through the forest, and I bump up against the Cordyceps mushroom and the perithecium gets stuck to me. And now I'm just walking through the forest shooting Cordyceps spores off of me. Like a little squirrel walks by and gets a bunch of perithecium in its fur, and now it's running around the trees shooting Cordyceps spores because they're active. Those little perithecium that falls off when they're ripe are actively shooting out spores till they die or dry up or whatever. So there's like at least an hour running through the forest on a squirrel or on a bug, shooting out spores everywhere. And I've witnessed them shooting out spores under the microscope. This is what they look like. So these things just fall off of the cordyceps, and inside of them, it's lined with the acini that the spores are produced. So it literally looks like somebody sitting at the end of the tip here. I don't know if you guys can see my mouse, but it looks like there's like somebody operating a turret of spores, like a machine gun turret of spores just shooting out of there, sometimes underneath the microscope. So you can only imagine that landing on some moving animal or bug or something like that in the forest and traveling. So these what the spores look like on the left here. They look like little shards of glass. They're straight lines. On the right is a bunch of perithecium that the spores germinated out of. So if you leave the perithecium there long enough, a bunch of spores will just germinate in them and grow out of it. You can also get clones this way, so it's not efficient for breeding, but it's really cool to look at. So the process for me for cultivating cordyceps, since I breed cordyceps, it starts in the wild. So I go out and I have to go find parent strains in the wild. So we do this generally like June, like late May through, not even May. May's way too early. I would say like mid-June if we're lucky, and into early October if we're lucky. So they have a pretty decent season. So we go out and get as many as we can, as many diverse specimens as we can, different morphologies. We had some friends find a white albino one, and my buddy up in Michigan bred out an albino cultivar of Cordyceps militaris. So we go out and look for all of these interesting variations, growing in different habitats, showing different morphologies, growing on different host species. There's 2 different host species here. On the left, we have Anisota centaurea. In the middle, I don't know what this little brown one is. This little brown reddish pupa. I have no idea what that is. But you can see the mycelium just like busting out of the seams on these little insects. This is the type habitat that we find Cordyceps militaris in: hemlock dominant forest with oak understory. Moving water they love moving water more. Rarely I'll find them far from moving water. But I will find them at the top of mountains sometimes, but I generally find them in valleys and streams with hemlock dominant. And I think that the reason why is the same reason why there's other more medicinal and sensitive herbs and other mushrooms in the hemlock forest, is because the leaves don't fall off of the hemlocks. So in the wintertime, the forest floor doesn't get oxidized and hit with solar radiation all for like 5 months, which is very damaging to life. That takes a different type of life to exist in full sun during the winter time. So I think that the hemlock protects a lot of very sensitive organisms over winter. We'll find them in the moss. We'll find a lot of them in the moss. A lot of people will say only look in the moss, but you'll find them in the forest, coming out of the ground and coming out of the leaves, and they move depending on the time of year. So like one year I saw them at the creek, on the creek sides, all at the beginning of the season. And then as the weeks moved on, it was like they moved up the mountain. And then the next year they were in a whole different place. They were not even in the same place anymore. And it's because they're on insects that are moving. So I learned this from a nut guy, this guy named Bill Whipple. I was living in North Carolina, Asheville, North Carolina. And there's this guy, I would go teach at these organic grower school. And I would teach at the organic grower school. And there was this guy named Bill Whipple, and he had a nut collective of people that would go collect nuts, and they had a processing facility where it bring the nuts. So some people would go get like acorns and black walnuts and all these nuts. They would take it back to this place and make flours and cheeses and all this kind of stuff. And then they had people go sell it. And I was listening to a presentation he was doing one time, and he told me that the oak trees will not drop their acorns in the same place every year. He said one side of the mountain will drop, all the oaks will drop acorns for maybe 1 or 2 years, and then they will stop. And then miles away, trees that were not dropping acorns for a couple of years will start dropping acorns. And he says this diversifies the squirrel populations and other animal populations. If they would keep dropping the nuts in the same place every year, a lot of animals would homogenize and inbreed with themselves because they wouldn't have to go far away from where the food is. And I mean, that's like a human way of viewing it, but even if that's not the reason why the oaks are like somehow all in communication, or we're not dropping nuts this year, we're gonna drop nuts this year. Even if that's not the reason why, it's still result of that happening. It's still like because that happens, the diversification of squirrels and other animals happens because they have to go move to find their food after a couple of years. But with that understanding and knowing that the biggest host of the cordyceps in Pennsylvania is the oakworm moth that eats oak leaves, I'm thinking that there might be something similar going on, because we'll find the cordyceps in one location one year, and then the next year they'll be in a whole different place, yards and yards away, and not in the area where we found them before at all. So I'm wondering if they're moving with the oak trees, or there's still a whole pattern there that I haven't fully understood. I'm fully convinced that nature is a language. I'm fully convinced that DNA is protein syntax uttering itself into existence, and that I've been crippled by English in the way that I think. But if I can remember that I am nature and that is all a story, it's all a language, then I can get to the end of those patterns faster and understand what's happening. Because like I'm making assumptions off of preconceived notions of being an English person. Like I'm not an English person, but an English speaking person, indoctrinated into this culture and all these kinds of things. Whatever. I found that leaning on my roots in experiential knowledge and just like understanding that knowledge that's in our own DNA is even what helped me to understand how to cultivate these mushrooms. Because whenever I learned how to grow them, nobody that could speak English could show me how to grow them. There wasn't any guides on how to grow them in English. It was all in Thai and Vietnamese and Korean and Mandarin. I didn't know how to speak any of those languages. I knew a little bit of Mandarin, but just trying to just recognize, I always could recognize that that nature was symbolic, like a symbolic language, and leaning more on that and stop imposing my human view on it was more how I figured out, all right, the cordyceps wants to eat this, and it likes to be in this kind of light, and it likes, like, you have to get yourself out of the way to see those kind of things. But yeah, so we find them. This is one on a larva. Those are more rare specimens to find, the one on the left there. And on the right, that's our production manager for Mycosymbiotics. His name's Justin. He's an incredible forager. He's been foraging his whole life. I didn't even go in the woods really till I was like 18 years old. And he was like foraging morels with his family. They owned an apple orchard up on the mountain in southern central Pennsylvania near here. His family owned an apple orchard. And I don't know how many of y'all know, but apple orchards have the most morels in our area. Like, you can go out for days trying to find morels and all these different types of ecotypes, but if you go to the apple orchards, they're just all right there. So he grew up picking morels, and he has a different perception of the forest, and I really value his knowledge. But yeah, so he's been helping out, and we go out foraging cordyceps, and we'll find lots of them. When we're foraging for breeding, we keep them in these bead boxes, and we'll bring like Q-tips or little soft bristle toothbrushes, or these little squeezy air blower. I don't know what the correct tool name is for it, but it's used to blow dust off of camera lenses. It looks like a stress ball with a straw on the end of it. But we use it to blow the dirt off the cordyceps, so that we don't, because if you put them in there with dirt on them, the dirt will get all over the spore producing parts, the peritheecium on the top. And that's what we need when we take them into the lab. We want the spores. The spores are invaluable. The cordyceps has a dollar value. As like a wild product, there's a dollar value on the market, but the spores are invaluable. If you can breed them, there's no limit to how much you can make off of that. this is a day of harvesting cordyceps out here in Pennsylvania. And that's like not even putting a dent on it. That's like not even going but a couple yards, because you'll get into some good patches where they'll be like all around your feet and stuff. You don't even have to move that far. As soon as you get down on the ground and you're picking one, then you can see another one while you're down on that level. And then you see another one. Like every one you stop at is like a beacon. And you have to really think about it like that. Like anytime I go into the forest and I find any kind of mushroom, like if I find an oyster mushroom in the forest, I stop and then I start going in a radius around it. Mycelium grows out like a circle. It's all coming from a nucleus somewhere. Like somehow that oyster mushroom got there, unless it's by the creek and it got washed down there in a big log. There's a nucleus of it somewhere. There's a big cluster of it somewhere. That's where it came from. So like every time you find a mushroom in the forest that's not a mycorrhizal mushroom, like a saprophytic mushroom, there's a big cluster of it somewhere that the spores came from. There's like a mom nucleus or like a mothering nucleus of it. So like I always try and like find the radial points of it. Like where did it connect from the last? How did the spores get here? And that usually puts you in a good position. I mean, like most mushroom foragers end up doing that without knowing. Like you find a maitake, or I'm gonna walk around this area, and then you find another one, or I'm gonna walk around this area, and then you find another one. Instead of just walking in a straight line, you can start navigating from spore drop to spore drop. I feel like that's what it is. Like you get down there, you see one antenna, you can beacon to another little one, you beacon to another little one, and to another little one. So yeah, if you don't want to go out into the wild and find mushroom cordyceps, which I don't know why you wouldn't want to, it's so fun. You can buy parent strains, which is another great thing to do, because like then breeding cordyceps on top of that is like a whole task and a half, and it takes technical skill and time and energy, all this kind of stuff. But if you want to just grow some, you can buy strains from people like Terrestrial Fungi, people like Appalachian Gold, or people like myself at Mycosymbiotics that have taken the time to breed out commercial genetics. So that you can grow out really nice strains. So what I do is whenever I get home, I take the cordyceps mushroom, I break it off of the insect, and I adhere it to the top of a petri dish with Vaseline or super glue. And I'll do this also with cultivated cordyceps mushrooms. While it's hanging over the petri dish, and you can see there it says WA on the petri dish, that stands for water agar. And that's just water and agar. That's a liter of water and 25 grams of agar powder. And I use this non-nutritive agar so that the spores germinate very slowly. This gives me time to isolate them from each other before they connect with each other. So you can see on the right is that little line in the center is the spore, and the mycelium grows all out from it like that. And I have to catch that mycelium before it touches another mycelium, or I have to catch that isospore, that ascospore before I have to isolate that ascospore before it touches anything else. Because as soon as it touches one of those thread touches a thread of another spore, it's on sexual reproduction, and they already exchanged their mating types. And then it's already bred out. I can't. There's nothing I can do with it. It'll probably produce some genetic. But if more than two of them connect, then you get multi-spore cultures, and it produces a bunch of different kinds of looking cordyceps, which is really not good for commercial production, because if I'm growing at a commercial scale and it gets into real business and it's all numbers and you have to produce the same amount and you have to meet some whatever quota, you got to put 5 pounds of rice in, you got to get this much out. And if I'm growing a multi-spore culture, one 5-pound bag of rice may produce, like, I don't know, I'm just throwing out random numbers. 100 grams of cordyceps in one bag might produce 200 or 180, because they're throwing out different size cordyceps, throwing out all sorts of different looking cordyceps. But if I get two spores together, they're going to produce all the same looking cordyceps mushrooms. It's all going to have an average harvesting yield. And I based my harvest production where I would like my production to be for my strains off of pint jars, because that's what we were cultivating at first. So if it doesn't produce at least 3 dry grams of mushrooms per pint jar, it's not commercial for me. So I don't want to deal with that. So this one on the left is not a single ascospore culture. So sometimes they'll connect before I can isolate them. And the reason I know that this one on the left isn't one is because it has little mushrooms growing around on the mycelium already. So I'm just like, it won't grow mushrooms if it's a single spore. On the right, there we have the single spores, and they turn yellow with light pigmentation, and they'll start to turn orange with more pigmentation. That is a really good sign for me. I mean, like, I associate that pigmentation with beneficial compounds. Like if not just the pigment. Like if I take multiple cordyceps cultures and I put them all in the light, and one gets super dark orange and one just stays white, I want that one, the super dark orange. It just made a bunch of something that the other one just didn't. And I don't know what it is until we get all the analytical equipment and all that, but I can see with my eyes, I can taste with my mouth that one just did something that the other one didn't. So we'll get a lot of these spores isolated. And then I'll determine the mating type by running PCR. If anybody is interested in doing PCR or is already doing PCR, you can go to minipcr.com and use the code MYCO5, MYCO and the number 5 for 5% off all their stuff. That's our special mycosymbiotics code with them. That was a really, really great plug, getting affiliated with those guys. Just to keep doing this kind of work without, as an independent, you know what I mean? Like, working together is amazing. Working with institutions is amazing. Working as an independent gives you a lot of freedom, but it is expensive. We'll determine the mating type of the isolated ascospores. And this is the gel that I was talking about before without any visual on the left. So for every 2 little holes at the top, can you guys see my mouse moving? Yeah. Okay. All right. So for every 2 of these little holes on the top, there's 1 ascospore culture, and each one of these holes has a different mating type primer in it. Primers will make sure that you get a specific gene when you're doing DNA amplification. So this first one is a mating type 1. I got a result in the first well here, but I didn't get any result in the second one. This little line down here is just blank, nothing. So this one tells me this is a mating type 1. This one, these 2, only the first one lit up. That's a mating type 1. These 2, nothing happened. I might have like a little faint result here, but I didn't really get a good result from that one. This one, again, mating type 1. Nothing came in the second one. So either 1 or 2 will light up. If both of them light up, it means it already was mated. So I'll combine the opposite mating types in a dish together. So I know this one's mating type 1. I know this one's mating type 2. I'll take a little piece of each of them and I'll put them together on a petri dish, and I'll intentionally let the mycelium touch. When it touches, it combines, it breeds, and then you have new strains. Then you have to test your new strains. So I'll test my new strains in the fruiting room. Cordyceps fruiting rooms are a little bit more forgiving than other grow rooms because they don't necessarily need to be held at a specific humidity. Really what you need is light and AC. You need to be able to keep the temperature at 65 degrees all the time, and get some high power lights in there, and you're good to go. Like, I like the T5 full spectrum fluorescent lights for growing cordyceps. And after you grow them, then you note the traits of the offspring, and maybe go back to the drawing board and breed different parents with each other to get certain traits, or you maybe just find something that you really love in the first breeding round. So you see all these different variations. Like, these are cordyceps. Like, I went out in the forest in Pennsylvania, got these mushrooms, got spores from them, put them together, and got all these different looking cordyceps growing on the same substrate, in the same jars, in the same grow room, and they look all different. Those 2 have one of the same parents, and they look so different, just like me and my brothers and sisters. And like, I like the length on the one on the right. I like the way the ones on the left look, like with the bulby tops and the bumps, the peritheecium on there. But yeah, so you have to note all these things, and then you end up with lots of cordyceps, and then you pick them and consume them. And this one made me feel like this one made me feel like that. And go back and combine the isolated spores with desirable traits, and then we sell the cultures, and people can grow them all over the world. So why do we love cordyceps so much? There's a lot of interesting species-specific unique compounds in cordyceps that make us feel good. Cordycepin is very similar in composition to adenosine triphosphate, which is used in our cells as the most basic unit of energy. And because it's so similar, it can go into our cells and provide us with a similar energy that our body recognizes. Cordymin is another very interesting compound. Cordymin and cordycepin have been shown in research over the past 10 years to, on their own and in conjunction, inhibit HIV-1 reverse transcriptase, which is like, whoa, crazy. Like this natural medicine is able to inhibit the HIV virus from replicating. So, there's so much more to unpack with that. And I do see it having a bright future in as an adjunct therapy for sexually transmitted infections and diseases, because a lot of these compounds seem to be very beneficial for other STIs and retroviruses. Other retroviruses would be like malaria. HIV is a retrovirus. So this is very interesting, and through the pandemic, we did see a couple research papers come out showing that cordyceps could be effective against. I don't know. I'm on live on the internet. I don't know what I'm allowed to say. I keep getting banned off of social media. They keep blocking me for saying things. So I don't know what I'm supposed to say nowadays. But I know we're on private. I'm just being a clown. So yeah, the cordyxanthins are more of the pigment compounds that are species specific, and then we have the cordycepine and the cordylan, which I'm less versed with their specific attributes, but all together in conjunction lead to quite the desirable experience. A lot of people have been utilizing cordyceps to kick their amphetamine habits that culture pressed on my generation. I don't know how many of y'all are of the 90s generation, but I remember going to school with kids that were on Ritalin, pulling their hair out and biting their cheeks off and stuff like that, and like really just going at it on their fingernails and like all this kind of stuff. Like that's crazy. Like a lot of my friends and I like really thank my parents for keeping that away from me. But a lot of my friends had to grow up on amphetamines and like had to feel like, one of my best friends told me he would go to school feeling like there's worms crawling under his skin and stuff like that. It's crazy that we have a generation that grew up like that. And so I've had a lot of friends in my generation tell me that they've been able to use cordyceps to get off of those things. And that's incredible. Because as somebody that's not taking them, like I don't even drink caffeine really. Like I never took those things when I was younger. And if I drink like a mate, I can't drink coffee. I'll be crazy. I'll be talking faster than I talk right now. And I'm like, I'm off cannabis right now. Like I have to suppress a little bit for how energetic I am. Like life to me, I don't know how people got so not enamored. What's the opposite of that? I don't know how people got so jaded with life. We're literally living in the most magical experience ever. It's a lie that we're not living in heaven. Like it's so magical that we can breathe and communicate with each other and go see these things in the nature and eat and drink. All these things are amazing to me, and people are so unamused by life. It baffles me. I don't even know where I was going with that, but I'll just get back to the cordyceps. So yeah, bag technique. We've transferred this more into like using a sustainable silicone bag to sterilize our material in, and then transfer it into a bigger container. And then we're able to fruit our cordyceps out in bigger containers now. These are excerpts from my book, which is available at mycoshop.net or mycosymbiotics.com. Here we have our commercial mix. This is something that I developed after 2 years of looking at and I don't even know, countless research papers on cordyceps. There's so many research papers out of Asia on cordyceps, on getting the mycelium to hyperproduce bioactive compounds in liquid culture vats. Um, a lot of people are like so interested in producing mushrooms, and then there's a lot of people that are interested in just the compounds that the mushrooms are producing. If anybody's interested in just the compounds that the mushrooms are producing, you don't need to grow mushrooms. You can just grow vats of liquid culture mycelium and extract the compounds out of liquid culture. You can do this by growing vats of liquid culture mycelium or jars of liquid culture mycelium, however you want to grow it. You can stick a whole gallon of a glass gallon jar that you can buy at a local brewery supply store. You can stick one of those into the big pressure cookers. So you can pressure cook a whole gallon of liquid culture, grow your mycelium in there, filter it out in a strainer, and put it into a freeze dryer. If you're so lucky to have a freeze dryer, I've been trying to get one for a while. But you can freeze dry it, you can dry it somehow, and then you can extract the medicinal compounds from it and do whatever you want with the medicinal compounds. And that's way faster than growing mushrooms. You can grow liquid culture mycelium, so many pounds of that stuff on a regular basis, and where it takes weeks to get to the point where you can actually get the mushroom to grow out of the substrate. So by looking at all these liquid culture recipes for hyperproduction of compounds in cordyceps, I was able to deduct different things and experiment with these different inputs, and I dialed it into this mix for our solid state substrates. And in my book, I go into why I picked all of these different things and what they do. Like peptone turns into cordycepin by breaking down into nitrogen, and cordycepin has a nitrogenous base, and all these things is broken down in the book. So it tells you how the cordyceps is digesting or whatever and turning into what. But really, you want to make sure that it has a nutrient-rich substrate, because insects are very nutrient-rich. Insects are nutrient-dense, more than a lot of the foods that we're eating out of our grocery store. Just being honest, like if you go out into the forest and you got a pound of beetles and then a pound of regular beef that's not organic or not grass-fed or whatever, I guarantee you that the insects are way more nutrient-dense than that beef, and way healthier for you, and not filled with antibiotics and not all that other stuff. Because they were outside foraging the minerals from the trees and all of the nature that can actually get nutrients, not some field that doesn't have any nutrients in it. So yeah, rice cooker and instant pot technique. This one's really easy for beginners. You can kind of pasteurize your substrate in the rice cooker and then put it and scoop it into containers after it's pasteurized in the instant pot. My buddy Ryan has a really good YouTube video on instant pot technique. Jar tech is how a lot of us started. It's really reliable. I mimicked this method from watching a lot of YouTube videos from Thailand. I saw them growing in jars, so that's how I started doing it. And yeah, been through a lot of experimentation. Dang, my hair is so long compared to that. Shoo, that was a while ago. That was back in 2017, 2018, when I was operating a cordyceps farm in Weaverville, North Carolina. And that was at Pisca Gourmet Mushrooms and Brevard, North Carolina. They let us use their big autoclave. So I started experimenting with doing Rubbermaid containers and bigger containers. So when I first started doing with bigger containers, this was before it went all open source on the internet. I was just trying to sterilize the rice and the substrate in the container, because we'll put the liquid with the rice, dry rice, and then sterilize it, and the heat cooks it all together. So I was just putting the whole containers in there. Now we sterilize it and then just put it into a clean container once it's already sterilized. So we'll sterilize it in some sort of reusable vessel and then put it into a clean container. So yeah, lots of experimentation over the years. This is an example of a cordyceps farm in Asia. On the left, they got full AC, lights on every rack, like just maxed out cordyceps mushrooms, clear lids. That's a very good thing to note, because cordyceps are photosensitive and will grow towards light. So if you have them in a mason jar with a metal lid and the light's coming in, it's only coming in from the sides, the cordyceps are going to grow towards the sides. So when we have them growing with the metal lids, we have to turn them so that they don't grow just in one direction. If you don't ever turn them, they'll just grow towards the light. It's pretty crazy, like a plant. Here's some of my first experiments with cordyceps cultivation back in 2016, 2017. Trying them out in different materials. They grew really well in the aluminum trays, but a lot of people were concerned that it would accumulate aluminum. So I didn't want to be selling people stuff that they would be fearful of. I haven't done the analytics yet. I'm literally, thank the Lord, liberated to do research. Like I have been grinding. When I started doing this, I was serving tables. I was a high school dropout serving tables with a baby at 20 years old. Like there was no and I'm brown in rural Pennsylvania. You know how tough it was to make money out here? It was crazy. People like already had all sorts of preconceived notions about me and all this stuff. So like, especially with having like only a GED and all this stuff, everybody's like, I was a statistic. Literally, my whole life, they tell you you're a statistic, all this kind of stuff. 1 in 3 black men will go to jail. I was that 1 in 3 black men, like for regular stuff that everybody's doing. So it's just like, it's crazy to be growing up like that. So like coming from that and serving, I was literally serving tables, like taking any little scraps that wasn't going to my kids' diapers or the bills to try and grow some mushrooms. And it went from that now, knowing all this stuff, knowing I'm capable of like, I'm like 18, 19 years old, knowing that I know how to do science and know how to do all this stuff, but everybody looks at me as a young black kid that doesn't have a high school degree and has a little kid. That's how the world looked at me. And I knew I could do this science and I knew I could do DNA. Not only I knew I could do molecular biology, I knew I could teach myself molecular biology. So I had to go prove the world to that. I finally figured out now I'm liberated to do the research that I wanted to do. I just had to keep working to make money, pay the bills when I want to be doing research. So like everything that you're hearing about, all this stuff I know about cordyceps is information I gleaned while I was just trying to grow the cordyceps to sell them to make money, because that's what I had to do to even get to this point. Like there wasn't no point where I could just sit back and do some research. So like now we can do the research. So all the points in time where I was like, oh, well, we didn't have the analytics, or oh, we couldn't do that. Now we can do that. So thank the Lord that we can do that, because that's amazing. And we're about to learn so much more. I know my time's about to run up, so I want to just show you guys some cool pictures of some of the cordyceps that I've grown over the years. This was my first breeding trial. I was inbreeding them because I didn't, like the first time I got the cordyceps, I only had one specimen from the wild. I cloned it. We were able to grow it. I tried to share it with a bunch of people on the internet. Ryan Paul Gates from Terrestrial Fungi in Michigan was the only other person that grew them with me. But we were able to get them to grow. And I took those spores and bred it back to itself. And I just kept inbreeding them and trying out and experimenting with how to figure out how to get them to grow all at the same time. I can't even tell you, like off that last little rant, like back then it was so crazy knowing what I had in my hands, like knowing that I was like knew how to grow cordyceps, everybody, and like that there wasn't even a market value for it. When I first started growing cordyceps, I was able to sell them for $1,400 a pound dry in the United States because I was the only one in the United States that had it. And like people are like, are you gonna patent this, or you should trademark and da 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 da? And I was like, nah, I'm gonna write a book and sell it for $10 on the internet so everybody can figure out how to grow these things. Cause like, what am I supposed to do? You know what I mean? Like something could happen to me. Why am I gonna hide this stuff? Like, and then it went crazy. But now, even now, you can sell cordyceps. If you're just a regular person, you can sell cordyceps for $600 a pound. If you have a good business, you can sell for $800 a pound dry. That's more than cannabis prices in Northern California. That's crazy. That's good business. So yeah, I implore people to grow cordyceps mushrooms. So some more cool pictures of our cordyceps growing. This is cordyceps over Astor Place in New York. When I first started growing them, I used to dry them on paper towels. Eventually we got into like drying them in dehydration, forced air dehydration. And where I want to go is freeze drying, just to preserve all of everything that we can. Here's one of my students' farms. I started teaching about this as soon as I started learning. I've been teaching people how to grow mushrooms since I was 21. So for 6 years, I've been teaching people how to grow mushrooms. I've taught over 10,000 people how to grow mushrooms. And I've been able to visit my students. I've been able to see people grow their businesses and all this kind of stuff. So it's one of my students' farms. up in upstate New York, and they were having issues not being able to grow cordyceps because after they left my farm, they started putting the heat in because they started listening to somebody else's direction. They started putting too much heat in, and that could really mess up your cordyceps. Here's one of my experimental grows in Weaverville, North Carolina, where I was just trying to figure out how to get it done. And we produced lots of really beautiful cultures. This was the 003 culture from Mushroom Life back in 2018, 2019. These were some of the first big harvests, made me a happy boy. And definitely started pushing me in a good direction. This is like a little bit of the social permaculture. As I mentioned, I'm convinced that life is a language, and I've been very interested in how I was sold crap. Like, how did I grow up as a young man listening to this crap music, trying to want to live this crap life, chasing fast cars and all this nonsense that was sold to me? And I was like, why is this so appealing to me? It's like primally appealing to me. And I was like, there's something linguistic about this, outside of understanding words as language. So I started to use those aspects, incorporate them with different, healthier environments and habits. And it's been successful. Because like I remember, I don't know, I want to keep talking stories. I don't know what over my time limit. I'll tell stories another time. But the social permaculture has been successful in infiltrating media. Last year I got put into all these crazy media outlets internationally, millions of views by intentionally wanting to put permaculture and good stuff into the mainstream media. A lot of people get into permaculture, get into the good stuff, and they don't want anything to do with mainstream media. Some people want it too much for the wrong reasons. All I wanted to do was to make sure that there was good videos on there, because I was the kid that was on the internet watching the videos, and there wasn't the good videos on there. And I needed it to be appealing to me. I needed to see somebody cool like me that could give me a good example of something good I can do with myself. Everybody that looked like me that was doing something or successful was doing something bad. And I wanted to make sure that the kids that grew up next didn't have to deal with that or had something else instead of that. So we've been successful in doing all that kind of stuff. And then we started doing cordyceps extracts. So I got into cannabis before I got into cordyceps, and I was always wondering why people left the alcohol in their extracts, because when people do cannabis extracts, they take the alcohol out of it to make like hash or concentrated cannabis extracts. So I started doing that with cordyceps. And collaborated with other businesses and started doing wholesale of cordyceps concentrated extracts for value-added products for different companies that are making like edible products or capsules or all sorts of different topicals and things like this. Concentrated extracts, like these things like are very bioactive. And yeah, I've been experimenting with this for years, and I'd love to talk about that more, but that's like a whole different talk. So yeah, these are the compounds in cordyceps. This is cordyceps minus, like the cordyceps fruiting bodies. We grow them, we wash off these beautiful oils and fats and flavonoid compounds with ethanol and water, and then we remove the water and the ethanol, and we're left with this, and it just smells like buttery popcorn. It's so crazy. I don't know. Do you guys want to answer some questions or anything like that, or ask or answer some questions? Because I'll just keep going. Like, this is cordyceps growing on cacao. I did this back in like 2018. I don't know why I won't let me go back. I did this back in 2018 because I was studying the fermentation process of cacao or chocolate. And I was like, why don't I just intercept the bacterial fermentation and yeast fermentation with cordyceps and just do fresh cacao into cordyceps fermentation, which worked. And I was able to even fruit cordyceps off the cacao beans instead of a bug, which was really, really cool. And the beans lost all their bitterness, and I was still able to make like chocolate. Like we were able to mill it down and make like, chocolate with it, which was really fun. I don't know if you can see the mycelium through it. I can't say for sure that because chocolate's already very stimulating for me. So I can't say for sure if it was like also enhanced with cordyceps. Here we have some teas. Back in like 2018, 2019, we found that cordyceps with vitamin C, not vitamin C, with cordyceps with a little bit of acidity can increase the amount of compounds that comes out of the mushrooms. We're cooking with them for a while. There's a big pile of like lion's mane and king oysters and cordyceps. Do not eat the mycelium after you've grown the mushrooms on it. That's way too long of the mycelium growing. You can eat cordyceps mycelium, but not after it's been sitting in a jar for 50 days. That's how long it takes for the mushrooms to grow. That's like a long time of rice sitting in a jar. Just eat the mushrooms, not the rice. Here we have cordyceps in cold water. And my friend Reishi did research back in 2018 at Bastyr University. We set up a research project on different extraction methods of cordyceps mushrooms, and she found that fresh fruiting bodies of cordyceps will yield high amounts of cordycepin and other bioactive compounds into cold water. Here we have fermentation with pickles, cordyceps pizza over there, ramen with cordyceps mushrooms, just different ways of infusing and getting cordyceps into your body. Cordyceps popsicles with cold water, fresh extraction with the fruiting bodies in there, so you can like be eating the popsicles and then chew up a little cordyceps mushroom while you're at it. We have a lung tonic that we did with other herbs that are good for lung health, because cordyceps is pulmonary, beneficial for pulmonary system, beneficial for lung health. So that was another fun project. And again, I could talk about all this stuff forever. That's what I was saying. Some chocolates. There's a lot of research from Asia showing that chickens fed cordyceps waster medium or mycelium can increase yolk size, decrease cholesterol, and also infuse their yolks with cordycepin. So you can have designer eggs. And yeah, there's on the left, cordyceps cephalocola. Ophiocordyceps cephalocola grows on wasps. You can utilize as a biopesticide. On the right is Talipocladium ophioglycoides, which could be utilized, can be grown in the same methods like Cordyceps militaris, and that one is beneficial for prostate health and also has neurological benefits. So yeah, those are the old harvest days back in 2018 in North Carolina. Just been teaching people since. Yeah, I hope you guys enjoy the presentation. I know I went a little over time, but I appreciate you guys listening to me today. You guys can check out more of the work at mycosymbiotics.com. We have a lot of videos on Apex Grower. I'm on Instagram at mycosymbiote, and check out MycoFest. MycoFest has its own Instagram, has its own website, mycofest.net. First weekend of August every year, August 5th through the 7th this year in Tamaqua, Pennsylvania. 3 days. Experts in mycology from all around the country. We got Alan Rockefeller coming out again. Who else do we have coming out here? I can't remember who's all on our confirmed speaker list, but we got a lot of really cool heads coming out this year. Let me see. If you guys have any questions, feel free to ask.
We'll get to Q&A in just a second, but first of all, I just want to say thank you for just an amazing presentation. It's super inspiring. And thank you for sharing all that knowledge, that's taken you years and years and years to accumulate, and you're just like spreading it back out there, which is just fantastic. So if you read the chat, you'll just see a lot of people going like, keep going, this is great, just don't stop, keep going with the stories. There was also a bunch of questions, and I was just going to call on people to ask the questions themselves. So I'm going to let people unmute themselves. And the first question came from Ashley Denio. You actually had two questions, but why didn't you talk about your speakers first before we get to Q&A? Because you got some really cool speakers there.
Yeah, these are just some of our confirmed speakers. We have other confirmed speakers. We have Michaela De La Maico coming out. We have Ryan Paul Gates from Terrestrial Fungi up in Warren, Michigan coming out. We have Mariah coming out from the Pacific Northwest with her plant medicine. Alan Rockefeller, if you guys don't know him or haven't had him talk for your club yet, please do. He's really great.
We've had, he's great, yes.
Reggie Harris is coming out from Oakland as well with Oakland Hyphae. They've been doing a lot of work in the psychedelic mushroom scene for the regulation for the whole state of California. K-Dubs, the hiking scientist, she joined us last time. She's really good at doing forays. Franklin Pozo, we have David Poplin also coming out from California. California is going to be representing hard this year. Robin Gunkel down from Baltimore. Jasper Degnars is coming from Guatemala with Fungi Academy. to teach about mushroom education, mushroom cultivation. Dustin Powers is coming out from Washington to teach about permaculture. We also have why is my brain speaking right now? Probably because I was just talking for so long. Josephine from Eco Agaric in Uganda is going to be coming out and doing a demonstration on how they cultivate mushrooms in Uganda. Yeah, it's going to be super, super fun. And this year, I was able to launch the International Mycoliteracy Scholarship. So we're able to fly out one student from the Global South and provide them full ride to the weekend before. We have a 3-day cultivation class on how to cultivate gourmet medicinal mushrooms. So we're flying them out for that. And then they get to stay with the team for a week and see how we operate our business here. And then they get to stay for MycoFest and get to learn from all of the other speakers. So we're really excited about that. And we're specifically picking an educator from the Global South so that they can take the knowledge back and share it with the people where they live.
That's fantastic. What a great initiative. Thank you so much. So now it's time for Q&A. Ashley, now it's your turn, if you don't mind asking you 2 questions.
Sure. Let me just scroll up so I can remember how I worded this. First of all, William, just super big inspiration from one kid from the 90 to another kid from the 90. You make a lot of good points. You have a lot of very relatable perspectives, and I appreciate your perspectives in the field of mycology as well. My first question was, are there any specific features that you've noticed or that you know are documented of insects or hosts that Cordyceps seem to associate with? Because to me, it seems like a lot of somewhat random host, but then again, I'm not that well versed in Cordyceps.
So where we live, it's mostly moth pupa. Like rarely do we see them on beetles here in the mid-Atlantic, or really, I have not seen any documentation that I can recall of people finding Cordyceps militaris on beetles in North America. There is documentation of it that, like in research papers and stuff like that, but most of the documentation on beetles is from Asia. So we're mostly finding them on moth pupa here. And from my understanding and my experience, I'm seeing them more as like a biological control mechanism. I think that they're keeping the insect populations from eating the whole oak populations or whatever other trees. Because like some years you'll see them more on one species and less on other species, and I'm just like, damn, like
Interesting.
Yeah, and they won't wipe out a whole population of insects either. So there's definitely some level of consciousness there, or whatever
absolutely.
you want to call it.
There's some behavioral decision there.
Yeah.
And my other question was for someone who is interested in PCR but doesn't. What would you recommend as far as an avenues to learn more about PCR, specifically maybe at a low cost level?
YouTube. That was what I did. Yeah, during the pandemic, I just watched lots of YouTube. That's when I learned how to do PCR. It was just like lots of free time. We were all stuck at the house, and I was just watching YouTube videos.
Do you know of any channels that you would recommend?
Alan Rockefeller.
You know what else too? Oh, Alan Rockefeller got deleted off YouTube. His YouTube got taken down. Okay. Yeah, they're really, the censorship lords are over here going crazy. I will put together, I've been working on some blogs. I'm going to make a note of it right now to make a post on our new website of resources for PCR education. Because I'm working on a book on it right now, because there's not really, for like, it's been so ivory tower that all the information on it is like ivory tower. It's not like ready user-friendly stuff. But now that there's like mini PCR and other companies come, like, I mean, I was about to say Oxford Nanopore, but they're definitely not user-friendly like mini PCR. But definitely like companies like mini PCR coming out, and companies like Omega Biotech that have user-friendly consumables are making it way easier, but there's not the literature or anything like that there. So I just gleaned information off YouTube. I'll get resources together, and do you follow on Instagram or anything like that?
Yeah. I'm on Instagram. I
Cool,
think
cool.
I recently messaged you on there. Something about, what's it, a message?
Right on. Well, I'm gonna make a post about it because that's a good thing to
Awesome.
share.
Yeah, I'll look for it. Thank you very much.
Yeah, my pleasure.
Okay, great. Next question from Melissa. Do you want to ask a question? Melissa Emma.
I forgot that I even asked a question. Where?
You asked a question, can you tincture the mycelium?
Ah, yes. Sorry, I have ADHD and my executive functioning stops at this time of the night. So, yeah, I'm just wondering, you were talking about, yeah, because I tincture, I'm an herbalist. So I do a lot of tincturing, and I liked what you were going to get into about, burning off the alcohol and being left with just the concentrate. So that really interested me.
Yeah, I've been doing that with success with lots of different. I forget. I think my code is Symbi for it, but there's a company called Extract Craft. I'm pretty sure my code is Symbi, SYMBI. But if you go to extractcraft.com, they have multiple different products for home ethanol recovery. So it's like a little unit you can just set in your kitchen. It looks like a regular kitchen appliance that you put your tincture in it, and it concentrates your tincture and it gives you your alcohol back. So you can like you use the alcohol again.
There's a home water distiller. I think that's what people were. I was watching a YouTube video and that's what I saw. So I know yeah, that's cool. Thank you.
Yeah, that's pretty cool. But and you can tincture the mycelium. Just make sure it's fresh, because if it's growing for too long, it can harbor other organisms.
Yeah, you said that towards the end of the talk. I remember. Don't eat the rice, eat the mushroom.
Yeah.
All right, the next question is from Erin Coughlin, the Paris. Do you want to ask your question?
Yeah, sure. Thank you for such an amazing talk. It was just a question about what you said about the mushrooms growing towards the light, and why might that be? It seems, somewhat unusual or different because they don't photosynthesize.
They're photosensitive. Their insects are in the soil. I believe that it may be one of their mechanisms besides gas exchange of finding out where this top is, or gravitropism, but just to see where to grow up and out. Growing towards the light when you're down on the forest floor, it ensures that your spores are up out of the ground. So I think that might be what it is. Just trying to grow towards like up high, towards you get your spores up further away from the dirt.
That makes sense. Yeah, thanks.
Yeah, that's just my assumptions.
Okay, great. Ros Lowen, you had a question.
As Cordyceps ascospores break up into part spores, but your cultures showed like a whole long spore. How did you manage to keep it together?
That's the way that I collect them, with the Vaseline on the petri dish like that. The spores just gently drop right onto the agar.
Oh, okay.
If I was to harvest them any other way, like there's like people online on YouTube that posted videos about breeding cordyceps, and they say to do liquid dilutions of spore dilution. So like just collect a bunch of spores in water and then put less water and less water, or take a drop out of that and put it in a thing of water, take a drop out of that, put it in a thing of water until there's less spores in it. But by doing that, it breaks it into part spores, which I don't know. I don't know if it's less valuable, but for breeding, I just want to have the whole thing. I don't want to be losing any piece of it. I don't know. Because from their breeding processes in using part spores, people have been able to breed out cordyceps that are producing mushrooms, but I just rather not lose anything.
It's interesting. Just a comment. This morning, I watched a video. I think David Wald hosted somebody named Danny Miller, from the Pacific Northwest, and he gave a lecture on how to do DNA studies, including PCR, and it was really very, very well done and easy to understand. So if somebody wants to learn how to do DNA work, that's one way. It's available. I found it on Facebook. So it is available if you're interested.
The fungal sequencing group on Facebook, great place to find information. Fungal sequencing on Facebook.
I'll drop that into the chat. And I think somebody's just unmuted themselves who shouldn't be talking. Sorry, I'm trying. Okay. Okay, sorry. We're good. So the next question is from Andre Dominguez. Andre, do you want to ask your question? Andre? Okay, so I'm going to ask you a question for you. For foraging cordyceps on the East Coast, any lookalikes to be aware of?
Clavaria or Clavulinopsis is the only thing that looks like it, but it's not like dangerous.
Mm-hmm. Great. And then Brittany, you had a question. You want to ask it?
Yes, thank you. First, I just wanted to say I'm so excited that you're doing MycoFest in Tamaqua because I'm from that area. And I was waiting to hear where it was going to be. So I'm really excited about that. But anyway, what kind of a gel do you run it on? Is it like a native gel or like a denaturing gel? I'm just starting to learn about gels. So
for gel electrophoresis, I run it on agarose, 2% agarose. I get my agarose from Mini PCR. It comes in tablets.
oh, okay.
Like a blister pack of agar tablets, and I just add water and put it in the microwave, and then I pour it into that little slant. Like, they're making it so user friendly. It's amazing.
That's awesome. My other question was just like, if you're like, let's say you're backpacking somewhere in Appalachia and you come across cordyceps, but you're not going to be home for a few days. Is there like a good way to keep it with you till you get home?
Take the dirt out. Don't dig the bug out. Like, dig a circle around it and pull the whole thing up with the dirt with it. And then wrap it in a wet paper towel.
Okay.
And then try and keep it from
And
getting
keep the towel wet.
like keep the towel kind of moist and try and keep it from bumping around. Like, try and keep keep it, like, if you can put it into like a coffee cup or something like that, so it doesn't like bump around or something, that would be the best. And then yeah, just give it some fresh air every now and then. It's like it's an interesting finicky game to try and keep a cordyceps from dying once you pull it out of the ground. If you're like not around human stuff, like if you're going to be backpacking and you don't have like a refrigerator and like all that kind of stuff, it's a tricky game, but you can do it.
Awesome. Thank you so much.
All right, and I think that was the last question. I just posted a link to the talk that Ross mentioned into the chat if anybody's interested. And this is it, I guess. Thank you so much for taking the time to not just, tell us more about cordyceps, but to answer our questions. It's really very much appreciated. You've got a very inspired audience here. So thank you.
Thank you so much. I mean, I can't believe you guys, that was like one of the biggest turnouts for a mushroom club talk on Zoom I think I ever had. So you guys rock for that one. And I don't know when you guys are doing your forays again, or if you guys are doing forays this year, but I'd love to come out and join you guys on one.
Okay, well, the New York Club does walks every single weekend of the year. So if you're ever in New York City, you're more than invited to come along. We'd love to have you.
Nice. I might take the Amtrak up sometime.
That'd be great. All right. Well, thank you.
Thank you, guys.
Have a lovely weekend.
You as well.
Everybody. Okay.
Thank you. Have a good weekend.
Yeah. Good night, everybody.